ايش اللي صار بالنسبة للstudy that was published yesterday. This is actually the study. It's not published yet. Had a website that has preprints of biomedical papers. فالstudy title is Uncanny Similarity or Unique Insights in the Novel Coronavirus to HIV-1, GP120 and GAG proteins. الauthors من University of Delhi, and I will tell you how they reached this conclusion. وحقيقة في كثير comments على ال non-scientific word uncanny, which is not really appropriate. But anyway, let's go to NCBI, which I'm sure most of you use عشان to search the literature. So we will go and search for the Wuhan sea market coronavirus. And if we search for this, we will identify طبعا novel coronavirus 2019 in COVID, and you click on this to see the code, the genomic code of this طبعا مع references. This is the protein code. Go down عشان نشوف الجينوم. This is the genome. So genome طبعا بالفور letters اللي احنا بنعرفهم goes down to here. It's 29,080 800 أو 900 almost يعني حوالي 30,000 nucleotide. This is an RNA virus. So similar طبعا ال HIV is also an RNA virus بس it's 9,000 bases, so هذا 30,000. وحقيقة شيء مذهل إنه فقط بهذا العدد القليل من ال viruses من ال nucleotides we have so much complexity of the virus. Anyway, so we saw this, and the first thing that scientists do when they have something like this is to run a blast. So we'll go and run a blast. What is a blast? It's a software that will do something called local alignment. So it will try to identify هذا الجينوم طبعا اسم الجين, and we will search for all viruses, the organisms with state viruses, and will simply blast. So what will this will do? This will look for all organisms that have, or viruses, so we specify viruses that has similar genetic code. So anyway, so if we do this, it will take a while to search and to refresh itself until it finds, yeah. And this is the result of the blast. This is what you will see. So the virus aligned very well to other different isolates. So we'll see. وبعدين أقرب واحد بصير البات SARS like coronavirus genome, وهذا السيميلاريتي 89%. الاي فاليو هاي مهمه جدا لانه it tells us how significant. This is very low, so it's almost zero. It's very low, so this is extremely significant. This is if we want really just to look على alignment of one of those viruses, let's say this one مع البات SARS like virus, and just check the actually let's just see how the graphically these look like. So كل خط يمثل genome of one of the viruses. So those are the Wuhan viruses لحد هون. This is the Wuhan virus. بعدين بيجي عندنا الباد SARS like virus. على طول we will notice إنه في gap هون. في part of the genome موجود في new virus that was not present in the bad. So this is an area of heavy research. We can look specifically at this and select one of these strains as I said. Go back, look at the graphic summary, and again we are looking at this region, look at for alignment. وهذا بحط لنا الكود of the virus, the queried virus اللي هو new coronavirus مع other virus اللي هو that SARS like virus. I'm gonna actually the first alignment is aligning the Wuhan virus by itself. So I'm gonna skip to the second virus. Yeah, here we go. So here now this is aligning مع path SARS like virus. على طول نبدأ نشوف إنه في فروقات because of mutation. This is an RNA virus. So this is very expected to have some differences here and there. ما في مشكلة. المشكلة تيجي لما يصير فينا insertions. هون فينا insertions. هذا insertion يعني هذا النيوكليوتايد موجود في الكويري فيروس بس مش موجود في اول فيروس. في عندنا insertion. هذا طبعا similar insertions 1 2 nucleotides ما في مشكلة. If we are looking for طبعا I can't do this manually but something like this يعني يكون في عندنا multiple insertions. So this an area like this. طبعا هذا هو النيوكليوتايد or RNA sequence. But if we translate it to amino acids, we may see insertion of amino acids here. So هذا هم اللي عملوه في الستدي. They identified هذول Let's go back and just check الريجن اللي احنا حكينا عنها. Let's go down 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 and check. طبعا هذا الفيروس مع بوقف ب 21685 في another aligning segment. احنا عارفين انه في segment missing. Anyway, so في another aligning segment that has الجاب اللي بينهم extends between 21696 and 2375 almost 1300 و 1400 bases مش موجودة بالصح. So again the scientists will go and blast this region because they want to know what it is, so they will blast it. And I did this before, so I hope I have the result. Yeah, here we go. So if we blast it, it will align only a short segment of this region will align to another virus. See the name of this virus. Here we go. So ما في any other virus, only this bad coronavirus فيها في alignment. E value is very significant. Estimated 78% في هاي السيجمنت. This is just this segment اللي فيها similarity. The rest of this is not similar to anything. وهاي هي المشكلة. So if we look at this, there's nothing, no similarity to anything. وهذا هو اللي خلى الساينتست to suspect إنه في something here. وهاي الاريا is يعني heavy research is going in this region. Okay, so see, yeah, this is the region. We can look at the sequence, but to study it further, what people do is try to do something called yeah, we go to something called ORF finder. So this is open reading frame finder. We will use الكود الموجود في هاي المنطقة. We'll try to find is there any protein that can be translated from this region. start codon or end codon. So it will look for any start codon or end codon عشان يعطينا open reading frames. On this region we've identified 5 open reading frames. الموست امبورتنت one هذا هو ال OR5 اطول واحد فيهم 351 امينو اسيد وات overlaps مع السيجمنت اللي موجوده بال البات SARS like virus. So they go and they blast this region وبعدين بنعرف let's skip this لانه it will take a little time. Here we go. Yeah, so this region is really similar to the spike glycoprotein in another bad coronavirus. So a segment is not totally new, but definitely as a whole, the segment is not identified. But see, when you look at the protein structure of يعني, within this segment, you can see it is very similar actually. 94% to another protein in another coronavirus. So this is still يعني within biologically acceptable evolution of the virus, we might say. طيب. Okay, so we'll go back to this study, and they did not really look at the spike protein in the listen حكينا عنه. I think they looked more at the inserts. So this let me just show you the table. This is the table that made. So they identified 4 areas of inserts زي ما حكينا موجودين بالفيروس موجودين بالSARS coronavirus, and then they queried. What they did simply they blasted those regions. أخذوا short reads وهي مشكلة كثير لما only 6 amino acids and you blast them. When they blasted the first read the first so-called insert, they got this. So they got, this is similar to an envelope glycoprotein, the human immunodeficiency virus, with 100% similarity, but the e-value is extremely high. This is 174. So it tells you, this is most likely this is by chance. وهاي المشكلة إنه they did not report an e-value. They did the same for the other proteins, and they got really they did not find the HIV. I mean, HIV was there, but maybe number 65. A very high e-value, similarity to other viruses. So really, it, it's not clear how they reached their conclusion. But then, obviously, they did another round of blast. طبعا هذا مش blast, the blast B. Um, but they specified, let me see what, yeah. So they specified in an organism called a human immunodeficiency virus type 1, and then they did the best they see they found the code in the HIV virus and the other codes in the HIV virus, but this is all with very high e values. So this is all not significant, and because they did not report their e values in their study, and it all appears to be happening by chance. يعني the chance, finding six amino acids or short runs of amino acids by chance is there, and with this very high e value, it is very likely that this is all by chance. So now it's considered, this paper is falsified, and the results are not really considered to be of any scientific significance.